Good afternoon, everyone. For those who don't know me, I'm Jackie, and I'm the next group member in Clive Mark's research team looking at arsenic as a potential cause of marsupial decline. So my specific project is looking at contemporary marsupial exposure to arsenic in the environment. But first, I want you guys to all look at this photo. What do every single one of these little animals have in common? That's very true. But you know what I think they have in common? I think they look pretty tired. Tired of what, you ask? Extinction. Since European settlement, 25 Australian mammals have gone extinct. That makes up 50% of global mammal extinctions. Of these, 10 of them have been marsupials, with 1 in 4 marsupials currently threatened today. So it's clear we have a bit of an extinction problem with marsupials in Australia. And there's been lots of research to see what kind of things could be causing these declines. 5 main theories make up most of the explanations for why they could have gone extinct. The first one being habitat loss through land clearing and degradation. Second is hunting, such as the bounty program placed on thylacine. Third is changed fire regimes, which happened when Indigenous Australians had planned land burnings, and when colonisers came, that obviously changed. Fourth is invasive predators, such as red foxes and the domestic cat, which typically prey on small to medium-sized marsupials. And lastly is disease, which is what we're all focusing on. So there's been lots of theories to what this disease could be, because in the early 1900s, at the time of rapid population crashes, both the Tasmanian tiger and the Victorian eastern quoll showed a strange distemper-like disease. The symptoms of this disease included skin lesions, hair loss, altered behaviour, and a suppressed immune system. So many theories have tried to explain what this disease could have been, but none of them have been very substantiated. An unexplored hypothesis is that this disease could have been caused by toxic metals or metalloids in the environment. Specifically, it could have been arsenic. At the time of these declines, arsenic in Australia was very widespread, used across all states. It was mobilised through gold mining, used in agriculture as a pesticide or insecticide, and this resulted in over 1,000 highly contaminated sites across the country. However, arsenic use ceased in the 1950s, so it shouldn't be a problem anymore. Unfortunately, it could be. The Environmental Protection Authority did a paper in 2016 looking at arsenic in Victorian rivers, and what they found was astonishing. They found some Victorian rivers had 110 parts per million concentration of arsenic, which is 10,000 times the World Health Organization's drinking water guidelines, which means this could be having implications for marsupials and conservation today. An example of this is this little guy, the eastern barred bandicoot, who had a previous range across the southwest of Victoria. So there's been lots of reintroduction programs going on, most of which have been largely unsuccessful. So this has been blamed on the red fox or poor management. However, we found that these sites crossed over really strongly with gold mining, which would mobilise arsenic in the west of Victoria, which means if arsenic is affecting contemporary marsupials, this could have had a role in why these reintroduction efforts were not successful. But how do marsupials actually uptake this arsenic from the environment? Well, I've identified 3 main methods from the literature. These are drinking water, the soil matrix, and from their diet. Once it is ingested, arsenic has a range of toxic effects and disrupts many cellular processes. I've just picked 3 examples, but there's many more. So arsenic disrupts oxidative phosphorylation. For those non-biologists in the room, this is cellular respiration, or it's the cell's main method of creating ATP or energy. So if arsenic disrupts this, this means the cell can't produce as much energy and can sometimes lead to cell apoptosis or programmed cell death. Arsenic can also cause an unfolding protein response by binding with some of the thiol functional groups in the protein. This results in changed protein shape, which can change signal transduction in the cells and increase homeostasis regulators. Arsenic also reduces DNA repair ability, meaning that cell restoration capacity then reduces. If ingestion of arsenic is continual over a long period of time, this results in chronic arsenic poisoning, which, when at high concentrations, can result in cancer or even total system shutdown. When it's at lower concentrations, chronic arsenic poisoning can result in a suppressed immune response, a stiff, uncoordinated gait, skin lesions and scabbing, behavioural impairment, reduced weight and altered bowel patterns. All symptoms that cross over really strongly with the distemper symptoms witnessed in the thylacine and the eastern quoll that I discussed earlier. So what do we actually know? We know that arsenic is in high concentrations in the contemporary environment. We know that arsenic causes harm to mammals. However, what we don't know is whether marsupials are being exposed and whether they are uptaking this from the environment. Which brings me to my research question. Are marsupials exposed to arsenic? And does this correlate with arsenic concentration in their environment? So my project has two main aims. Firstly, I need to find out if contemporary wildlife are being exposed to arsenic and whether we can quantify this. So I'm going to do this by sampling fox hair collected through the Victorian Bounty Program. I'm sampling hair because, as Izzy discussed, hair is a common biomarker for arsenic. It actually gives you a good idea of how much arsenic that organism was exposed to throughout their lifetime. Why foxes? Foxes aren't marsupials. However, they are actually readily available to sample, as I can collect many samples from the Victorian Bounty and cover like a wide location around Victoria. So once I sample these fox hairs, I'll go to different locations all across Victoria to find these. Then I'll bring them back, and using inductively coupled plasma mass spectrometry, I'll analyse these samples for arsenic concentration to see how many foxes were actually being exposed to it. That will then bring me to my next project aim, which is to find if arsenic in hair samples of marsupials correlates with arsenic in the environment. So I'll do this by collecting a whole bunch of dead wombats. So I'm going to sample hair from roadkill wombats in high and low arsenic sites. Wombats were chosen because they have burrowing behaviour and the soil matrix is a common uptake method of arsenic, and they're a common roadkill species in Victoria. The high and low arsenic sites will be selected based on the EPA data, which identified areas in Victoria which would have high arsenic and areas which would have lower arsenic. Then once we find the roadkill on the road, we'll set up transects which will go perpendicular to the road, which is that way, and we'll sample the soil along those transects to get a good idea of whether there is arsenic in that immediate surrounding environment. Then we'll use ICP-MS again to analyse the hair and environmental samples for arsenic concentrations, and I'll take these results and see if I can correlate high arsenic sites with higher concentrations in the hair of wombats. So my study has a few different limitations and roadblocks. The first one being, what if you don't find any roadkill wombat samples, which is a possibility. So what I've done to do that is I've started contacting wildlife shelters and wildlife rescue groups to see if when they have a wombat pass away in their care, they would send me a hair sample and I can go to the location they picked that wombat up to sample the environment. My second is to try and get roadkill cleanup crews to assist us in either sample collection or just to tell us their schedule so that we can go right before they pick up the roadkill so we get maximum dead animals. My other option is if we find we're not going to find many wombats when we're on a road trip, field trip, sample other marsupials whilst on the field trip so we at least can bring something back to analyse. My limitation with the fox aim is that we won't get an idea of the demographic data of the foxes. So what you do with the bounty program is you only get the fox scalp. You don't get the sex, you don't get the precise location, but you do get the rough locality, and you don't get how old the fox is. So in order to combat that, I'll also be talking with fox hunters to get samples with known locations, sex and age. So my next steps are, as Isabel mentioned, we're going to train on the ICP-MS probably next week. I'll be going to collect fox hair samples also hopefully next week. And I'll continue contacting wildlife shelters and fox hunters. In May, we'll do our field trip to go out and find wombat roadkill in the low and high arsenic sites across Victoria. Then I'll analyse these samples with ICP-MS and begin data analysis. In June, I'll finish this off, I'll collate the results and identify trends. So bringing it all together, Isabel's looking at the thylacine as a historical case study for whether arsenic could have affected its decline. Myself and Saxby are taking a bit more of a contemporary lens, with me looking at contemporary marsupials and whether arsenic could be a risk factor to them, and Saxby looking at arsenic potentially being uptaken via plants. So together, we're looking at arsenic as a potential decline both historically and contemporarily. I'm hoping that my research will really shed light on some more potential risk factors that could be facing conservation today. It's not these guys who need to wake up, it's us, to all the risk factors facing animals today. Questions? Oh, no. Thank you.
You use the term mobilised arsenic. So you're suggesting, therefore, natural arsenic, because some Victorian soils are very naturally high in arsenic. It's already there. It's not human contamination.
No.
So is that what you're suggesting, that it's the mining and other land management practices that have made that arsenic bioavailable?
Yeah, that's exactly right. So through gold mining, I'm not sure of the exact specifics, but I'll give you the general gist. Through gold mining, when they dig up the ore, that there's like some process when they dig up the ore that then mobilises the arsenic that was originally very deep in the ground, that then makes it available to more like top soils.
Can I follow that up?
Yeah.
So what's your control? Are you going to have sites that don't have mobilised arsenic?
Unfortunately, no, just due to the fact that we really don't know whether, because it's all historically mobilised, so the data wouldn't tell us if it was mobilised through gold mining or if it is just there naturally. So we can kind of go to a gold mining area and I guess that that would probably be mobilised arsenic, but it's not something we could potentially be sure of. Of course, I'd be happy to chat with you after if you do have any ideas.
Go to a place where it's probably not been sampled again.
Yeah. Yeah, it'd be definitely a good idea. It's not something I'd really thought about due to there's not great quality data around that, but I'll definitely look into it more. Thank you.
Why are you taking soil samples near where the dead animal's found if you don't know that that's where, like, is it that they'll be within a home range? Like, is it known knowledge that that'll be within their home range? Like, why would arsenic in soil near where it's dead indicate the arsenic that it's exposed to in its lifetime?
Yep, so this is once probably another limitation of my study, because we'll be using the EPA's data, which gives a good idea of arsenic all around Victoria. So that's how we'll choose a site. So when we go to a site, we'll know that that is higher arsenic than others. However, testing the soil is just kind of making sure that there is arsenic still in an area where that marsupial could have been. There's no way of us really knowing where its range was or its movement patterns. But by testing the roadside arsenic, at least we know it's been to this site, it's definitely been here, so at least we can test that environment where we know it has been.
How localised is arsenic presence, as in if in a 10 by 10 metre square, is it likely that that whole space will be pretty consistent, or is there great diversity?
It kind of depends. So in some cases, it can actually be very localised. So we, I don't know, if there's like a mullock heap from a gold mining site, it's likely that the mullock heap would be very high and then 10 metres away, not very high at all. However, if you go downstream, so like a place where water could have washed the arsenic away from an arsenic source, arsenic could be quite widespread in that case. So it just depends on, I guess, yeah, like whether it could be washed downstream of the original source of arsenic.
Actually related to marsupials, but the 110 parts per million in Victoria, 10,000 times the like safe limit. Is that actually like definitely dangerous? Like if you drink that water, you sit, you die, like it's 10,000 times like really dangerous. How dangerous is that?
For people or for animals in general? Just generally? Well, the thing is that would be a one-off exposure. So if it's an acute you just drink it once, it could potentially not have as much of an impact. I'm trying to remember off the top of my head, but I think it was also the study found that was the highest in particular parts of rivers. So this is not the whole river by any means, or all of Victoria. That was the higher bracket of some rivers within the state. But once again, like if you did ingest it and you were ingesting it over a long period of time, this would, I can't like make any definite claims that it would probably have health implications, which hence is why we're looking into that.